So noni, Murinda citrifolia, is a traditional Polynesian medicine used to treat a variety of ailments. This botanical supplement has been documented as an immune system modulator, anti-cancer agent, antioxidant, asthma alleviator, and anti-inflammation agent in scientific literature. Additional cited benefits of noni include improved homocysteine and blood lipid elevation, increased joint health, increased physical endurance, help in weight management, and type 2 diabetic blood glucose level regulator. The need for research into this botanical supplement is high. Understanding the properties of noni will lead to better health supplement alternatives. With greater knowledge of what noni affects, more research can be conducted and eventually even lead noni into a mainstream supplement to potentially help a plethora of ailments, including but not limited to chronic inflammation, type 2 diabetes, oxidative stress, cancer, airway diseases, and asthma. This project was based off of my previous work, which was mainly looking at T cell diversity levels. As you can see in the graph, a surprising result occurred in that noni modified both participant immune systems strikingly similarly. Therefore, the purpose of this project was to investigate the reason for this, as well as to see noni's effects on proteins and gene methylation. Although noni's benefits are well documented, the mechanism of action within the cell is relatively obscure within current scientific literature. Therefore, this study aimed to investigate the effect of noni on proteins and protein pathways, as well as the methylation of gene and respective gene pathways. This study additionally investigated the effectiveness of noni on immune health through metrics of T cell clustering analysis of the immune repertoire. The sample size of the study is limited. Two related participants are used. The participants are siblings. One is a healthy 18-year-old with no underlying health conditions. The other is a 20-year-old type 1 diabetic. Blood samples were taken on the 1st, 3rd, 10th, and 15th day of the experiment. Day 1 is referred to as baseline 1, and day 3 is baseline 2. After baseline 2 was taken, participants immediately started taking unfermented noni at 1,000 milligrams per day, sourced from a company healing noni. At day 10, the noni trial, blood samples were collected, and both participants stopped taking noni. At day 15, baseline 3 was taken. Baseline 3 is the recovery period after noni, where participants had just stopped taking noni for a week. In order to understand the results, let's dive into proteins and methylation. Proteins are created by the transcription and translation of DNA. Proteins, such as inflammatory cytokines, determine the response of the body to stimuli, and in this case, pathogens, as well as aid in other processes. Proteins relate to other proteins in a pathway. Think of pathways as a sequence of proteins leading to a response. Pathways determine the actions within a cell, and by extension, functions in the body. Methylation is a single method of the body controlling gene expression. The term refers to the addition of a methyl group to a gene, which tightens the gene not aligned for transcription, and therefore the protein will not be expressed. However, in reality, it's more of a case-to-case basis on whether methylation actually decreases or increases the expression of a gene. The bioinformatics program Ingenuity Pathway Analysis by QIAGEN, or IPA, was used to assess protein amounts and methylation of individual genes, as well as gene and protein pathways. Specifically, IPA helped identify changes in inflammation, cancer, asthma, and airway disease through protein and methylation analysis. Luminex assay was used to check 65 protein factors using IPA during noni and baseline 3 separately, which were compared to the initial baselines. From the Luminex assay data collected, the results were plugged into the bioinformatics program Ingenuity Pathway Analysis. Through bioinformatics, the relationship between proteins and their respective pathways were identified. The noni trial, as well as the baseline 3 trial, were separately compared to the initial baseline in order to see which proteins and protein pathways changed during these trials. This is an example of a template IPA shows. The specific data is for the type 1 diabetic males noni trial. Top protein pathways affected include airway pathology and chronic obstructive pulmonary disease, interleukin-17 signaling, and HMGB1 signaling. Both pathways underlined in black are part of the inflammatory response, which was similarly identified in diseases and disorders most affected. The overlap shows how many proteins in the pathway present in the dataset are in the pathway. The pathways underlined were all downregulated, meaning that noni decreased expression of the proteins in that pathway. Again, in the type 1 diabetic males baseline 3, similar pathways are downregulated, as well as proteins and as well as diseases and disorders affected. For the normal males noni trial, the same pathways were affected with only small changes in overlap, which means that the top pathways affected are slightly different. Additionally, diseases and disorders affected were the same. The baseline 3 results of the normal male for protein analysis show similar results. An overall downregulation in inflammatory-related pathways, as well as airway pathology and chronic obstructive pulmonary disease being greatly affected. The major takeaways are inflammation pathways were significantly downregulated, inflammatory cytokines showed decreased expression, airway pathology was significantly downregulated, and the inflammatory response was significantly affected. So, here's an example of the pathway provided visually by IPA. This pathway, HMGB1 signaling, was identified as significantly downregulated, which in simplest terms means that there's just less of that specific factor in all participant datasets. Green signifies that there is a decreased measurement in the dataset. Blue signifies that the proteins that IPA predicted as downregulated. Orange indicates a predicted upregulation, and red means the factor was upregulated in the dataset. Each protein connects in the pathway to other proteins, leading to the regulation of pro-inflammatory proteins, which is at the bottom of the pathway, and lung disorder also at the bottom of the picture. As you can see, pathways can be incredibly complicated. However, for this presentation, we will be mainly focusing on what the pathway ultimately does. On the left hand of the slide, major functions of the gene pathway change through methylation are shown, which is also what is encased in red in the picture. Interleukin-17 signaling had significant changes in methylation. The affected functions of this pathway include inflammation, pro-inflammatory cytokines, and invasion of cancer cells, which were all downregulated. Methylation data of both participants was combined in order to see the sort of average effect of mammalian pathways. In case you miss it, I'll reiterate how methylation affects gene expression. Although methylation can result in less gene expression, it doesn't necessarily always equate to it. In fact, it's more of a case-to-case basis on whether methylation will decrease or increase gene expression. Therefore, this data shows significant methylation changes in the pathway. However, cannot show whether the pathway had increased or decreased expression, such as in the protein analysis. The NONI trial for methylation showed cancer as top disease affected, however, ultimately did not reap as many results as baseline 3. The major takeaways are that cancer was significantly affected, and pathways related to oxidative stress were affected. Baseline 3 showed changes in cancer pathways, as well as asthma and airway disease. Cancer was similarly the top affected disorder. The major takeaways are from this baseline 3 is cancer pathways were significantly changed, and cancer was significantly altered. The pathway IPA provides for genes works identically to protein, except instead of looking whether there is more or less protein, these ones are looking at greater or less methylation. For example, green means that the gene was less methylated in the dataset, etc., etc. In this pathway, the senescence pathway, which is cellular aging, changes in methylation had occurred. The affected functions include cellular senescence, proliferation of cells, and cell division cycle. In the FAT10 cancer signaling pathway, the affected functions through changes in methylation include tumor growth and cancer cell invasion. GAD45 signaling also had significant changes in methylation. The affected functions include the cell cycle and apoptosis. The molecular mechanisms of cancer pathway also had significant changes in methylation. The affected functions include the cell cycle, apoptosis, and cell cycle regulation. So, from the visual pathway, many functions related to cancer have significant changes in methylation. For example, cellular senescence, apoptosis, proliferation of cells, cell division cycle, tumor growth, cancer cell invasion, G1/S phase transition, D2/M phase transition, D2/M DNA damage checkpoint were all significantly affected. As a reminder, changing the methylation changes expression, ultimately changing the response of the body. We also observed significant changes in asthma and airway diseases. So for this pathway, airway inflammation in asthma, we can see that acute severe asthma, Th2 immune response to the lungs, recruitment of eosinophils, and recruitment of M1 macrophages were all significantly affected. Interleukin-17A signaling in airway cells was also significantly changed in methylation. The affected functions include inflammation. The role of interleukin-17F in allergic inflammatory airway diseases also had significant changes in methylation, and the affected functions include airway inflammation, neutrophil recruitment to the lung, and allergic inflammation response. Airway pathology in chronic obstructive pulmonary disease, which you may recognize from the protein pathway data, was similarly affected. The affected functions include pro-inflammatory cytokines, alveolar cell death, as well as the degradation of the extracellular matrix. From this visual pathway data, we can see that many functions related to airway diseases and asthma also have significant changes in methylation. For example, inflammation, airway inflammation, neutrophil recruitment in the lung, allergic inflammation response, pro-inflammatory cytokines, alveolar cell death, acute severe asthma, Th2 immune response of the lungs, recruitment of eosinophil, recruitment of M1 macrophages, and degradation of the ECM were all significantly affected. Now, we also observed some changes in immune system cells. For example, T helper cell differentiation had significant changes in methylation. Additionally, 4-1BB signaling in T lymphocytes was also significantly changed in their pattern methylation. And the affected functions include T cell proliferation, T cell cytotoxicity, as well as activation-induced cell death. T cells are one of your body's primary defenders in the immune system. They detect pathogens that are specific to the receptor protein that they use to identify them. This protein receptor that is used to identify the T cell's specific pathogen is called the CDR3. Epitopes are what the receptor recognizes from the pathogen. Whole blood samples were collected, and packaging tube samples were shipped to iRepertoire for processing using the RepSeq analysis after every baseline or sample point was taken. The results are data of a frequency distribution of unique CDR3s. These files are used for single and two-stage statistical analysis. FASTQ files from each sample received from iRepertoire were used to separately process DNA and subsequent VDJ, and VDJ refers to the different gene segments used to create the T-cell receptor. Annotation for the CDR3 clustering analysis using the programs ALICE and VDJ-DB to calculate clusters and epitope associations. T-cell clustering is the amount of T-cells with the same CDR3 or a CDR3 similar enough so that the protein produced differs by one amino acid. Both T-cells will detect the same pathogen, and therefore they are considered the same cluster. A T-cell with a Hamming distance of 1 has an amino acid that differs from the specific receptor by 1, while a T-cell with a Hamming distance of 0 has the same CDR3. Now, let's talk about what we found in both diversity and T-cell diversity. No observed changes in T-cell diversity occurred. However, the clustering of influenza A and Epstein-Barr virus decreased during Noni. Currently, we are looking at an analysis of influenza A. As you can see, baseline 1, which is the first baseline we took, showed a large cluster of T-cells. However, during Noni, the cluster in both participants decreased. Additionally, once off Noni, the clusters came back. A somewhat similar effect is seen in EBV. For discussion, these results indicate possible ways that Noni could be modulating disease risk, notably cancer, asthma, airway disease, or COVID-19. And these results are observed in DNA methylation and at protein levels, so think about all the inflammation pathways we saw in both protein and gene expression, as well as the airway pathology in pulmonary disease. That was also seen in both protein and DNA methylation analysis. The ability of Noni to modulate inflammation and interfere in airway pathologies indicate Noni could be used to mitigate cytokine storm during COVID-19 infection. Further research could check RNA expression, use a larger group size, and use cell culture experiments to evaluate how Noni is changing DNA methylation. So for my reflection, I said I had a lot of fun with this project, but more importantly, I met a lot of influential people in my life through this project that have really just shown me the value of teamwork, as well as let me learn the perseverance needed to conduct research. So I'm very grateful about that experience. I highly recommend staying in touch with your mentor, exploring topics you're only somewhat familiar with, yet still interested about, and really just having fun with your project. So I'd like to say a thank you to Dr. Mauna Kea, Dr. Perez David, Dr. Chan, and Dr. Petrell. Here are my references. Thank you.